Hello everyone, my name is Megan Weatherly, and on behalf of the ADSA Graduate Student Division, I would like to thank everyone for joining us for today's Pioneers in Dairy Science webinar. The webinar today is titled Bovine Somatotropin, Regulating Nutrient Use to Improve Productive Efficiency and Reduce Carbon Footprint, and will be presented by Dr. Dale Bauman. Dr. Bauman is the Liberty Hyde Bailey Professor Emeritus in the Department of Animal Sciences and the Division of Nutritional Sciences at Cornell University. He was born in Michigan and raised on a dairy farm. He received his undergraduate and graduate degrees at Michigan State University and the University of Illinois. His research focus includes the regulation of metabolism and milk synthesis, production of animal-derived functional foods, and the environmental impact of animal production. And with that, I would like to turn it over to Dr. Bauman.
Thank you, Megan. This is my first time doing a webinar, so we'll see how this goes. Thank you for joining the lecture today, and thanks to the Graduate Student Association for organizing this series. Let me just start in then. Here's the outline that I hope to cover in the next few minutes, and so I'll move right along. First of all, in historical context, what is the background on bovine somatotropin? And there were several key early investigations. In the 1928, I believe it was, Evans discovered that pituitary extract would stimulate growth in a prepubertal lab animal. And sometime after that, in 1933, two scientists showed that it also affected lactation in lab animals. Then in amazing studies for the time, Asimov and Krauss obtained crude pituitary extract and injected over 500 cows. These Russian scientists, you can see some of their responses there, and you can see how when you give the injection, milk production shot up and then came back down. Another key early investigations were by the English workers in the 1940s. And these investigations came about because there was a food shortage in World War II from the Germans bombing England. And so they wondered if rbST could in fact help alleviate that food shortage. And so they characterized many aspects of the biology. And they came to the conclusion, though, that the amount of pituitary extract available was inadequate to aid the national food supply. A key thing about these investigations and the ones that occurred following this up to the 1970s were they only tested it in well-conditioned, low-producing cows, no cows over 7 kilos of milk a day. And they used over-conditioned cows because they assumed that the pituitary extract would work better in cows. They thought it worked by stimulating fat mobilization. If they had higher producing cows, then they were afraid that it would cause ketosis because the animal would increase its milk production and need more glucose. So we came on the scenes in 1970s, and I'll try to show along the way some of our thinking. Shown in this slide is the cycle of life, and we were asking the question, how do animals differ? So you can see that animals consume nutrients. In the first process is digestion and absorption. So that's one place where animals might differ. Secondly, once they get those nutrients, they can differ in the amount of nutrients used for maintenance. They can differ in the amount of nutrients used to make milk in lactation or conceptus if the animal is pregnant. And they can differ in the use of nutrients for body reserves. And in turn, then, this tissue utilization comes back and affects feed intake. So we asked the question, where do they differ? And it turns out they differ in nutrient partitioning to mammary milk synthesis. And so right away we knew that was where we needed to focus. And so we asked the question, how is the tissue use of nutrients coordinated then to support milk production? How can the animal direct all these nutrients to make milk production while preserving animal well-being? And about that time, my youngest son was subscribed to Ranger Rick. And so the story in this particular issue of Ranger Rick was the elephant seal, the northern elephant seal, shown here with his pup. Mom weighs about 400 kilos, and the pup weighs about 25 kilos at birth. And the pup nurses for a 30-day lactation and gains at the rate of 4 kilos a day. So that at the end of the 30-day lactation, the pup is strong enough to be able to go to the sea, go to the ocean with mom and survive. Now the impressive thing is the mother, because in that 30 days, she doesn't eat or drink. So the question is, wow, how does she do that? She must have acute regulation occurring minute by minute, second by second, to preserve steady state processes in her body. But at the same time, she must have another type of regulation that allows her during this important lactation to direct nutrients for milk synthesis. And clearly, the ability to direct nutrients for milk synthesis is the essence of survival of the species. So as we began to work on this, then we crystallized the concept that really regulation involved two parts: homeostasis, that had originally been proposed by Cannon, involving acute maintenance of physiological equilibrium. Glucose is a good example where the body has impressive homeostatic mechanisms to preserve a constant circulating concentration of glucose so that glucose can be partitioned in the right direction for the different tissues. But we proposed then a concept of a second type of regulation we defined as homeoresis. And we define it as a chronic coordination of metabolism of body processes to support a dominant physiological process or state. The term homeoresis had been used earlier in the 1940s by a scientist named Waddington. And so we simply crystallized the concept that he had originated. This was done then by Bruce Curry and I, and we realized the key features of the homeoresis then was it had to operate on a chronic basis. Homeostatic mechanisms operate on an acute basis. Homeoretic mechanisms had to be chronic. And it had to simultaneously influence many different tissues and processes. And in thinking about the way this could be done, we realized that a key part of the mechanism had to be altering tissue response to homeostatic signals. So the lactating seal could still preserve steady state conditions, homeostatic signals preserve steady state conditions, but when lactation began, she shifted to direct a substantial amount of nutrients for milk synthesis. So we were off and running. Now, how do we test this hypothesis? Well, in thinking about which hormones had been suggested or seemed to have mechanisms that might fit our definition of homeoresis, we realized that many reviews talked about somatotropin having a confusing array of different processes that it affected. But that array of processes wasn't confusing to us because we realized homeoretic controls had to coordinate many different processes. So I proposed that somatotropin was a homeoretic control nutrient partitioning. Then I had to get enough somatotropin to test the hypothesis. And that wasn't easy to do. At that time, the only somatotropin available was through NIH. They typically contracted internationally to obtain pituitaries that they would contract to extract and then provide the different hormones to scientists. somatotropin, or growth hormone, was one of these hormones, and it they usually gave it only for tissue culture type of studies or to use in radioimmune assays. So I had to convince NIH that they could give me lots of this hormone, and that what I was proposing was really a key scientific study. And I was able to do that, and so for a period of several years, through the late 70s, early 80s, we were able to obtain about half of the bovine growth hormone that NIH had supply. I also then had to get the grant support, and at this point in time there really weren't any competitive grants in agriculture through USDA, and so I was able to convince NSF as to the critical originality of our proposal, and they provided the grant support. So you can see there a typical example of a study. We would see the increase in milk production when we injected daily bovine somatotropin. And once we stopped injecting, milk production fell. So quite a few students worked through this period. Colin Peel and Tom Frunk and Phil Eppert, Stuart McCutcheon, Wendy Kohak, just to name a few. But it was about this time that biotechnology came about, and initially several companies, perhaps almost a dozen companies, were actively looking to explore the possibility of generating bovine somatotropin by biotechnology. The first company to get the recombinant bovine somatotropin was done through a joint arrangement between Genentech and Monsanto. You can see in the left photo then an E. coli that has the bovine somatotropin gene inserted into it, and these solid packages within the E. coli are really the bovine rbST. And so in December of 1981, they were far enough along that they had enough we could use in an actual test. And so this is a picture of the first injection at Cornell University on December the 11th. I remember that well because they had spent several $1 million to get to that point, and I remember solubilizing the rbST and the knot in my stomach as I did so. Well, here's the results of that initial little study. We had enough to do 4 cows for 6 days, and we compared the recombinant somatotropin to pituitary-derived or natural, and you can see they basically follow the same curve. And so we immediately submitted an abstract that March and presented it next summer in meetings at the annual ADSA meetings. Well, that was pretty exciting. No one had seen a technology before that could cause a 10-pound or 5-kilo increase in milk yield just immediately. And so it was really exciting from that standpoint. It was exciting because it was really the first product of biotechnology that had made it to a potential agriculture application. I must tell you that when we started these studies trying to identify the possibility that somatotropin was a homeostatic control, I never dreamed that it would result in a biotechnology product. I thought rather it would be used in selection systems and applied kind of to the more traditional ways when you identify key controls. Well, because of the excitement then, just everybody became involved in these studies once somatotropin was available, once recombinant somatotropin was available. So in animal science, you see the list here, the types of studies that were done by scientists in universities, in industry and the government, veterinary clinical science, Epidemiology studies, food science looked at the product composition and the manufacturing characteristics, and the economists modeled the adoption rates and impact of size and type of farm. And so it went from, in the early 80s, almost my group would have been the only one presenting papers related to bovine somatotropin at the scientific meetings each year, to where about 1985 and thereafter you had whole sessions, mini sessions organized around just bovine somatotropin. There were quite a number of major questions, and I show them here just to make the point as to the extent of the work that was done. Could the large milk increases be sustainable? And the answer to that was yes. Bioenergetics, how did it affect the bioenergetics of maintenance and the partial efficiency of milk synthesis? And it turns out it doesn't affect maintenance. Voluntary intake, would the animal increase its voluntary intake, or would you have to increase the nutrient density of the diet to supply sufficient nutrients? And it turns out the animal increases its voluntary intake. Genetically superior cows respond. If the cow is pregnant, will she still direct nutrients to the fetus? Or does lactation win the game when somatotropin is used? And in essence, priorities aren't changed. If a first lactation heifer hasn't completed growth, she'll still complete that growth in the normal way, with lactation being secondary. And what would happen if it was given to poorly managed cows? Would you see adverse effects? Would the cow try to produce milk at all costs if she was receiving somatotropin? The answer is, if she's in a poor management situation, you don't have any adverse effects. You just get a lower response or no response. So there was a lot of publicity. Some of it was interesting. This is an article National Geographic did that had our picture of me and our research group, and one of the first cows that was in a full lactation study with somatotropin. Not all the pictures were that good, though. There were a lot of people, a lot of misinformation associated with those early days. So here was a Humane Society put out this picture that supposedly was a cow treated with pituitary-derived BST to produce more milk. That's amazing. I've never seen that. So let's go to the next section then and consider mechanism of action. How does BST work? And this was actually one of the key studies. This is in the Beltsville Energy Chambers, USDA Energy Chambers at Beltsville, Maryland. This is Henry Terrell. And so he was involved collaborating with us on quite a number of really key studies. And it turns out, to kind of summarize this a bit, it turns out that somatotropin is affecting a large number of different tissues. Some of these effects are indirect, and in particular those associated with mammary are indirect, and I'll talk about that in a moment. But most of the tissues, the effects were direct, and these tissues had receptors for somatotropin, like adipose and liver. And so somatotropin would bind to the receptor and have an effect with that cell type. We can kind of divide the whole animal mechanisms into two components. One is it increased the mammary gland's ability to synthesize milk. And it did that by increasing, better maintaining the secretory cells, so persistency was increased. And the second thing that happens is milk synthesis per cell is improved. So you get a greater milk synthesis per cell, and then you also maintain the cells for longer times. These effects are indirect, and they're mediated by IGF-I. And when I say they're indirect, in fact, the bovine somatotropin is binding to the liver, and it's really the liver that's producing the IGF-I that affects the mammary. The second part of the mechanism involves somatotropin orchestrating the metabolism of the different body processes. And this is really a coordination that involves many tissues and processes. and it really involves all nutrient classes: carbohydrates, lipids, proteins, and minerals. Let me illustrate that with a glucose example. The ruminant, of course, absorbs little glucose, and so it meets its needs by hepatic gluconeogenesis. And those needs are quite substantial. Over 3 kilos a day in glucose must be produced in these high-producing cows. rbST, of course, would increase the glucose demand because you have more milk lactose, all of those carbons coming from glucose, and you have more milk fat with the glycerol moiety and the reducing equivalence portion being derived from glucose metabolism. So how does the cow respond to this? You might guess, just like they did for early investigations, that if you markedly increase milks, you would in fact cause ketosis to occur. The cow would become hypoglycemic and you develop ketosis. Remember, the milk is increasing just over the first day or two after you give the injection, so it's very rapid. Well, through lots of different types of experiments, we were able to show, we and others then all working on this, we were able to show that with AB difference across the mammary gland, of course, you can demonstrate the mammary uptake of glucose is increased, and obviously the use is increased with the output of extra milk. AB difference techniques also demonstrated that you had an increase in hepatic gluconeogenesis and that muscle uptake and use was reduced, and that the whole body oxidation of glucose was reduced. The net effect of this then is these shifts in liver, muscle, and other tissues in the body are making more glucose available for the mammary gland to use. So the increase in milk synthesis is accommodated, and the animal still preserves glucose homeostasis. Well, with that simple example then, I've got 2 slides here that list some of the processes in tissues, major tissues that are affected when you give somatotropin to lactating cows. Mammary tissue we already talked about. The liver, you have increased rates of gluconeogenesis. And normally the rates are regulated acutely by insulin. And in this case, the ability of insulin to inhibit gluconeogenesis is reduced, and that then allows, even when the animal would consume a meal and some insulin would be released in response to that, the liver would still make more glucose because it wouldn't be responding as greatly to the insulin signal. Adipose tissue, reduced lipid synthesis in adipose tissue. If the animal's in negative energy balance and needs more energy, it'll increase the lipolysis rate. And this too is done by modulating the tissue's response to homeostatic signals that affect lipid synthesis, like insulin, or affect rates of lipolysis, like the catecholamines, adenosine, and the prostaglandins. And here's a few more. I wanted to just, these you really have to go look at a review and study them in some depth, but one interesting example is the minerals, because the increase in milk also means you need more calcium, for example. And of course, calcium is another nutrient that has quite a tight homeostatic regulation. And so what happens then with somatotropin is you increase the production of 125D3 in the kidney. That stimulates the intestinal cell to produce more calcium binding protein, and that allows for greater absorption of calcium to match up with the extra milk. Well, all these type of changes then are coordinated, and these are the same type of changes that occur when you have a high-producing, genetically superior animal. So exactly the same. So with that as the background, let's turn a bit background and mechanism. Let's turn a bit and consider some of the commercial experience. These are fermentation vats making bovine somatotropin. So the chronology of the BST story is that in November 1993, FDA approved Monsanto's formulation. I mentioned there were quite a number of companies working to see if they could develop a product with BST, and Monsanto was fortunate to have a formulation for the release of somatotropin that turned out to be the best one. If you think about it, a protein hormone, then once somatotropin would get in the bloodstream, half of it's broken down every 18 minutes. So it really needs to be there each day, and there never had been an implant type of system for a protein. We're dealing with a protein here. And so Monsanto discovered a way to make a lipid matrix that would allow a slow release of the formulation. So commercial sales began in February 1994 under the trade name Posilac. And the Posilac treatment supplements begin 57 to 70 days postpartum and then can continue every 2 weeks until dry out. And through a lot of different commercial studies, follow-up studies, they confirmed that you typically see a 4 to 5 kilo a day increase in milk production. Then kind of a key marker in the story of BST is on October 2008, Elanco purchased the Posilac business from Monsanto. They had already been marketing the Posilac in other parts of the world, so that this was a purchase of the U.S. Posilac business. And as of June this year, over 35 million U.S. dairy cows have received Posilac. One of the things that was of interest and was what would happen to animal health and welfare. In that earlier picture I showed you of Humane Society's proposed cow that had received BST, those type of pictures and claims were things that frequently led to wild stories about somatotropin's effect on animal health and welfare. There have, of course, been lots of evaluations, large-scale studies on commercial farms once it got approved. Lots of scientific reviews have been written on different parts of the animal health and welfare. And, of course, we have anecdotal reports from 20 years of commercial use. And, in particular, I might call your attention to a recent meta-analysis by Normand St. Pierre and a group of us that was just came out last month in JAVMA, Journal of American Veterinary Medical Association. Collectively, all these different evaluations provide no evidence of any unmanageable adverse effects on milk composition, udder health, reproduction, body condition, lameness, or longevity. I think this was always an interesting thing to me because some of these people writing articles that it would cause lots of adverse effects seem to think that farmers were a little bit stupid and they would use a technology that caused their animals to have problems, not realizing that, in fact, one doesn't achieve high milk production without having a healthy, well-managed animal. The overall conclusion, though, was that commercial management practices used by U.S. dairy producers are adequate for the efficacious use of rbST with no adverse effects on cow health or well-being. So what's the value of bovine somatotropin, recombinant bovine somatotropin? And to look at the value, you really have to look at it on a per unit of milk basis. And I've listed 3 things here. Number 1, productive efficiency, defined as milk output per resource input. somatotropin then reduces the resource input per unit of milk and reduces the waste output. So a gallon of milk can be produced with fewer nutrients and less animal waste. Environmental impact, it mitigates or reduces the carbon footprint, greenhouse gas. And then economic value, you get a positive return on investment. Of course, a producer wouldn't use rbST unless they did get a positive return on investment. That has generally been in the range of $2 to $3 return for each dollar spent on the technology and the extra costs associated with using the technology. So let's look at some of these in a little more detail. The production efficiency effect occurs because of what we call dilution of maintenance. And this cartoon is intended to try to illustrate that. Shown in the bar graph then is the animal's nutrient cost. The dark bar at the bottom represents the maintenance cost, and then the top bar represents the nutrients then are used for the synthesizing milk, and they would represent, in economic terms, a variable cost. Now, the maintenance requirement of dairy cows on a body weight basis today is exactly the same as it was for my grandfather back in the 1930s and my father in the 70s and 80s. So that maintenance requirement really hasn't changed, that fixed cost. And so if you increase the milk production, then the animal has to eat more to make the extra milk, but all of that extra nutrients goes to make milk. And so it's diluting the maintenance costs from very low levels, around 33% in 1944 of the nutrients that go for milk, to where today we're up in the towards 70 and 80%. So this dilution of maintenance effect then this fixed cost effect isn't only nutrients. It in fact represents all of the dairy operation. All the fixed costs in the dairy operation then are reduced when you have cows that produce more milk per unit, per cow. So let me try to put that in context then, what that means. Presently in the U.S., we're producing about 200 billion pounds of milk a year. And so if we assume that 10 billion pounds of that milk is going to be produced using cows that are receiving rbST, these cows would produce an extra 10 pounds of milk a day through this interval when they're treated from day 57 to 70 through the rest of lactation. What's the implications of that? So this would be what? 5% of the U.S. milk, total milk produced using cows that are receiving BST. What's the implications if you didn't use that technology and instead you produced that same amount of milk? Well, by using rbST or any technology that gives you 10 pounds per cow more milk, you need 96,600 less cows, less animals. And of that, almost 53,000 are cows and 44,000 are heifers. That also translates into 721,000 fewer tons of feed to produce that same milk. 156,000 fewer acres of cropland. It saves fuel that can be used to heat households, electricity, and water. On the animal waste standpoint, it means that 10 billion pounds of milk is produced with only 874,000 fewer tons of manure waste produced. And when you translate it then into greenhouse gas or carbon footprint, it reduces that by over 500,000 metric tons, which is equivalent to removing 112,000 cars from the road or planting 83.5 million trees. So these numbers then get to be quite large when you think of what they mean on an industry-wide basis. Dilution of maintenance. It aids productive efficiency, the environmental impact, and then the economic return. So let me spend the last few minutes talking about some of the opportunities and challenges. So Elanco is marketing rbST as Posilac, but you also see in the grocery store signs, our farms produce its milk without the use of hormones, antibiotics, or pesticides. This would be organic. Or you also see labels rbST free. So let's talk about that for a minute. I think public discussion of new technology is important and essential. However, it's ill-served if the focus is on any misinformation. And I think that shows a need for us to participate. We as scientists do more than just do the experiments and publish the paper. We have to participate in the public discussion. We have to be involved and just provide the facts so that this can be considered and evaluated. I indicated there were lots of misinformation examples. This is just a few of the claims. Claims were put out that it would cause 50% of the farmers to go out of business, that consumers consuming the milk would get cancer and AIDS, the milk would contain increased pus and antibiotics and be of low nutritional value, and the cows would have catastrophic mastitis, mad cow disease, they'd burn out, become emaciated, and die. And in fact, just before BST was approved, Humane Society put out half-page ads in several national magazines, Time, U.S. News and World Report, making these type of claims. So what actually happened then? Let's think about the safety. Because this was one of the first products from biotechnology, I think the first product might have been human insulin, but this was right there with human insulin. Lots of the medical and scientific societies were interested in examining the safety considerations, and so many took it upon themselves to evaluate the safety of rbST. Of particular importance were FDA and similar regulatory agencies worldwide, but also NIH Technology Assessment Conference was quite an important conference. The World Health Organization and the FAO, United Nations, provide a health evaluation that's looked at as key for much of the world. But lots of other ones, AMA, American Cancer Society, American Academy of Pediatrics, HHS for USDA, U.S. Surgeon Generals, lots of different scientific societies looked it over, and all without exception concluded that rbST use was, the milk from rbST treated cows would be safe for human consumption. The reason for that, kind of some of the key reasons are here. Somatotropin is a protein, and when protein hormones are taken orally, they are digested and broken down to the amino acids, like all other proteins. Interestingly, in the case of BST, it was also known already that BST was inactive if injected directly into the bloodstream of humans. That came about because in the 50s it was recognized that some forms of dwarfism were a deficiency of human somatotropin, and so they tried isolating somatotropin from cows to see if that could be injected and overcome that problem, and it could not. Also, there's no compositional changes in the milk, and we actually knew a lot about the biology of somatotropin in general because of all the scientific research. And so the human safety of consuming these milk dairy products from cows treated with somatotropin was declared by all these medical societies and government regulatory agencies. IGF was another one that was looked at very closely. And the reason it was looked at so closely is that IGF, it's a much smaller protein, and it has the same structure in mammals, in all mammals. And so the same IGF that's active in cows would be active in humans. Now, there is already IGF in milk. It's very low, and it's highly variable. But it was found that it was known that when you gave somatotropin, then you increased the blood circulating levels of IGF-1. But it was found that even when you're giving rbST, the concentrations in the milk still remain within the normal range. Other reasons that were involved in this evaluation was we actually secrete IGF-1 in our saliva and all the different gastrointestinal secretions that go into our GI tract. And this amount is some 40 plus times greater than the amount in 1.5 liters of milk. So the amount consumed in milk would be negligible compared to the amount we actually produce in our body, in our saliva and GI secretions. Similarly, if you compare it to our whole body production of IGF-1, the amount in 1.5 liters of milk is less than 0.1% of our daily production. Now, it is a protein, so you would expect IGF-1 would be digested in the GI tract, but it wasn't until more recently where we had recombinant IGF-1 or more studies were being done in humans, directly in humans, that was shown that, in fact, oral IGF-1 is not absorbed in studies with newborn children, newborns, neonates, or young adults. So rather, it's digested just like other proteins. So on a worldwide basis, some 20 countries then have registered rbST for commercial sales, but the safety part has been evaluated by 56 countries, and they've confirmed the use of rbST is safe for the human food chain. No country has put up a regulatory ban on rbST because of human safety concerns. There are countries like Canada or the European Union that have not registered rbST for commercial use, so it's not allowed to be used there by the producers. But even so, the dairy products from countries using rbST can still be imported. There's no restrictions on the import, no special labels needed on the dairy product, no exceptions. So it's recognized that it's safe. As I mentioned, though, you do see in the food store the organic milk labeled as produced without growth hormones, or rbST-free milk. This product does not contain the growth hormone rbST. When you see articles in the lay literature or you hear lay people talk, most often when they talk about growth hormones, they're thinking of steroids. And of course, that's a whole different thing than protein hormones. So for that reason, I tend to like to use somatotropin to avoid that confusion. But marketing on the basis of developing niche markets on the basis of production practices, of course, has become quite common. In the case of rbST, there is no test to tell if the milk came from a conventional farm that might have been using rbST or a farm that didn't, or an organic production system. Now, FDA allows labeling of rbST-free as long as the claim is truthful. And so that's often done by a third-party certification, or in this case, farmers have to sign an affidavit that they're not using rbST. However, the label claims are not related to any meaningful differences in the major components of milk, the hormone content of milk, because there are some trace levels of hormones in milk, or the fatty acid composition of the milk. So there's really no test that can be used to tell whether it came from rbST-free herd or an organic production system. So I'm about out of time. Let me give you a couple final thoughts. In an interview I did a while back for U.S. News and World Report, I did some calculations. You can use the projected population increase for the world, and just by comparing the area under the curve, you can tell that in the next 40 years we'll have to produce as much food as we previously produced in the history of humankind. That's amazing. At the same time, the land per individual is going to be cut in half because of the increased population and because of the commercialization of land. The UN FAO, of course, is quite concerned about this and has done quite a number of studies. And they estimate that 70% of this additional food must come from the use of technologies that increase the efficiency. And rbST is one of those examples. So use of rbST allows for a sustainable production of high-quality milk with fewer resource inputs, less waste, and a reduced carbon footprint. It provides a safe, FDA-approved management tool that you can use to produce milk at an affordable price. And finally, I can't emphasize enough, I think it's essential that we understand and discuss the role of technology in food production, and that we discuss the resource conservation and environmental consequences of our choices in food production. With that, I shall stop and thank you for your attention.
Where do you think that the future is headed with regard to rbST use? I mean, you've outlined all the specific important aspects about the product, so I was just kind of wondering, where do you think we're headed in the future with it?
Well, I think for a period of time, certainly it's a technology that's going to be used in, quite extensively used in the dairy industry. If you think about it from the standpoint, it's really one of the key controls regulating or regulating nutrient use, and part of the basis for, a major part of the basis for some of the differences among animals. We know at some point when we're selecting animals, and we have so much better tools to do that today, we'll get an animal that has all these controls maximized and you'll be at the plateau. We're not near the plateau. The top world record is what, 72,000 pounds of milk in lactation, right at 200 pounds of milk a day. So that animal had all the controls right. And it's not only then the genetics, but the management. So when you put those two together, as we improve the genetics and we improve the management, we'll eventually get to a plateau.
Okay, great. Thank you so much. I was also wondering, how did you and your group deal with all the negative backlash from groups like HSUS, and how did you wind up being able to advocate for the use of rbST during the initial developmental stages?
Well, I guess I wouldn't say I was an advocate, but I would say I was, here's what it does. And that kind of made you appear as an advocate when you're talking about people that had a lot of misinformation or negative claims. That was an interesting period. I was, during that whole period, I was chairman of the Board on Agriculture and Natural Resources for the National Academy. So we were dealing with biotechnology more broadly, even in the negative publicity about that. And so it's just a matter of, look, here's what the facts are. You can make any decision you want, but make sure you got the facts right. And that's really, I think, what our role is as scientists.
Do you find that it's difficult today to advocate for the product or to inform the general public about it?
I don't know that it's any more difficult than it has been in the past. Certainly when you have the kind of a tendency by many today to be anti-science in general, that makes it more difficult. But just the facts. That's what we deal with as scientists.
Right. Okay, great. Thank you so much. Well, I don't see any more questions, so if there's nothing else, then I would like to thank our speaker today for joining us and delivering a wonderful presentation. So thank you so much for that, Dr. Bauman. I'd also like to thank all of the listeners for joining us today, and we hope that you all learned something new and enjoyed the presentation. I would like to remind everyone that the next webinar will be on October 15th, featuring Dr. Collier, who will be speaking about the interaction of climate and dairy cow biology, so please be looking out for emails about that. Once again, on behalf of the Graduate Student Division, thanks everyone for joining us today, and thank you, Dr. Bauman, and we'll see everyone next time.
Thank you.